Okay, can everyone see that title slide? Yes, all right. So as Tim mentioned, I'm the kind of the state medical entomologist with Colorado Department of Public Health and Environment. Thank everyone for joining today, especially with this being off schedule. And today I really want to talk about both my career path, how my education and interests led to me being in this position, but then really go into how and why we do vector surveillance. Why is it important? And as Tim mentioned, I'm going to give you guys the fastest crash course in vector ID that you can build on in your other experiences this summer. So I'm going to start with my background in education, how I ended up as medical entomology at CDPHE, and kind of what my tasks and responsibilities are in that role. Then go into why surveillance matters and our different vector identifications. So originally from Utah, after moving around a lot, I went to Westminster University. Then it was Westminster College for my undergrad. This is a small liberal arts college without a lot of specialization in terms of majors and minors. So I majored in biology, minors in chemistry and public health. When I started at Westminster, I feel like a lot of students interested in health in general, I was actually pre-med. Within a couple of years, realized that I was interested in kind of the bigger picture, certainly interested in pathogens, but less interested in the human side of that. So I started exploring options in public health. Around this time, the university got their a public health program for the first time. So took advantage of that, grabbed a couple of courses as soon as they were available. Was able to participate in some travel opportunities to Uganda and Thailand. And that was my first real taste of both global health and doing anything with vector surveillance and control. In Uganda, I learned a lot about malaria transmission and the different ways that we work to control that. And in Thailand, I got a side of the arbovirus side of things with Aedes aegypti and dengue control. So at the end of my time at Westminster, um, I knew that I was more interested in public health and specifically in mosquito-borne diseases. I was still a little bit unsure if I wanted to be on the strictly public health side of things, like working at a health department, or if I was interested in the research components of that, looking at mosquito transmission and control efforts. So with that in mind, I found a program at Tulane with the School of Public Health and Tropical Medicine with their MSPH program. And with an MSPH, which is Masters of Science in Public Health, I took, classes in epidemiology and in outbreak control, in public health policy, but I also took classes in virology and immunology and parasitology. I got both sides of that coin that I was still torn about which side I wanted to end up on. During that time, I had a great chance to do an internship, like many of you are doing this summer, with the City of New Orleans mosquito, termite, and rodent control. So I got to assist with their surveillance efforts throughout the summer, and then also participate in some projects that they were working on. The biggest one that I was involved in was actually looking at the blood feeding habits of local mosquitoes. So we took the collected mosquitoes throughout the season. Any that were engorged, we tested for what host that blood meal had come from, trying to see if there were pattern transmission trends. I also had a chance to participate in kind of the bench side of things for the first time. And in that, we were looking at dengue virus, Aedes aegypti, vector virus interactions, seeing what components of the dengue virus during its maturation process, during cell infection, were important for infecting the mosquito, and if any of those led to increased or decreased transmission. So after 2 years at Tulane, I thought I was leaning more towards staying in the science side of things. I thought, like, great, I have loved the research I have been a part of. Let's continue this path down academia. And I attended University of Texas Medical Branch, which is in Galveston, Texas, right off the coast from Houston there. And here I was in Dr. Scott Weaver's lab. And his lab historically and primarily does arbovirus evolution. Lots of sequencing work, lots of animal modeling. And I came in thinking like, great, let me kind of round out my experience and my skill set. This will be a little bit different than mosquitoes, and I'll come out more fleshed out on the other end. But then Zika showed up. And with Zika, there were some really important questions about which mosquitoes were transmitting the virus, what were the important factors of transmission, and where we were seeing that transmission happen. So right back into mosquitoes I went, and I loved it. I had the chance to work in Brazil as well as with the Houston vector control. I also got to do a lot of work in the lab. The focus of my dissertation was vector virus interactions, studying which mosquitoes were competent vectors of Zika virus and which factors inside the mosquito and of the virus contributed to that transmission cycle, looking at different strains of virus as well as different species and strains of mosquitoes and looking at all of these different combinations and what made a difference in transmission cycles. Really enjoyed that work. At the end of that, I found a postdoc in Texas, and the game plan was to do more field work in South Texas, doing some kind of novel vector intervention strategies right along the southern border, getting a lot of fieldwork experience. But I graduated in 2020, and COVID had shown up. So again, kind of my plan took a left turn, and field work got put on hold by the university, and I ended up doing a lot of lab work. The projects we were involved in, thankfully, I thought were still really interesting. I was in Dr. Gabe Hamer's lab. His wife, Dr. Sarah Hamer, was also a professor at A&M, and between the two of them, they decided to examine possible infection and transmission of SARS-CoV-2 to and possibly from household pets of human cases. So really interesting project. Again, this involved a lot of evolving and emerging public health concerns. The questions we were getting along the way, as well as the research that we had to do, had to be changing every week depending on what we were seeing come out. Overall, I did enjoy that experience. However, unfortunately, my part in that project was the lab work. Because I had some containment experience at University of Texas Medical Branch through the Galveston National Lab, I really became the lab guy, as you can see in that bottom right picture. Lots of time in high containment, lots of testing animal samples for SARS-CoV-2. We also tested samples from local white-tailed deer populations, and even from flies and other flying insects in the homes of cases to see if they were possibly transmitting anything off of contaminated surfaces. Thank goodness they were not. So again, interesting projects, but this really kind of solidified to me that I wasn't happy just focusing on the lab side of things. I missed that bigger picture, that public health application. So I started looking at other opportunities to get back into that world. This included applying for CDC fellowships and internship programs, keeping an eye on local and state public health departments. And in 2022, just a couple years ago, ended up with Colorado Department of Health and Environment. So my role at CDPHE is ultimately to support and expand vector surveillance and control efforts throughout Colorado. That's a big task. What that breaks down to is training local health departments around this time of year between kind of March to May. I bounce around the state, training different health departments on mosquito collection and identification, making sure that they are ready for the surveillance season. We've also started tying more tick surveillance training into that, and really just supporting what they need at the local level. Because as far as boots on the ground, I'm not the one conducting the surveillance. The local health departments and their mosquito control districts are the ones actually collecting the mosquitoes and either testing them or sending them to us to test. So I need to make sure that they are supported and prepared in that. My job also involves a lot of public outreach. This is everything from working with our communications team to develop social media infographics and messaging strategies, to doing interviews for newspapers and local news stations. Already this year, I have done about a meet once every week for the last month or so. And part of that is because how nasty last season was. There's a lot of concern about this season. I'll touch on those numbers in a minute. Also, to make sure that both I and our local health departments have the support that they need, I have to work on identifying possible funding mechanisms and relevant projects that we can be a part of. Possibly the best example of that so far is the Rockies and High Plains Vector-Borne Disease Training and Evaluation Center, RaHP VEC. Through this, we have additional funding and support, not only to analyze the existing surveillance methods, but to, train young students and young professionals interested in that field to make sure that we have continuing expertise in our region. I also am the representative for CDPHE for things on the vector side at both regional and national groups and meetings. I was a little bit surprised when I got into this role how many different regional groups there are, which is excellent. I work with groups in Utah and Wyoming, New Mexico, Northern Texas, Kansas, regularly, both comparing surveillance numbers, seeing if there's any trends throughout the region, but also helping support each other. If we've identified a funding mechanism that others might benefit from, or we've started to take a unique approach to a surveillance strategy, we share that in the region to make sure that everyone is as prepared as possible. And then lastly, probably unfortunately my least favorite part of the job, but a very important part is the data analysis. And when I say my least favorite part, I don't love doing data analysis, but I like what we can do with it. Being able to map and image where we're finding mosquitoes and ticks and using that in public messaging, I really enjoy that piece of it. And to get to that piece of it, you've got to crunch through the data. So that's something that honestly, I did not focus enough on along the way. You will never go wrong with data management or analysis skills as you are going through your education and early career. I wish I would have more, and now I'm kind of catching up in that process. So, big picture, what does West Nile virus look like in Colorado? This is what West Nile looked like last year. So last year was our worst year for West Nile virus cases in Colorado since the virus first showed up in 2003. So our worst year in 20 years. This graph shows the human cases. Along the bottom, we see weeks of the year. And along that y-axis, we've got the number of cases. This is confirmed human cases. The bars along the bottom are our previous 5-year average. At worst, we typically see a little over 20 cases a week. And then look at that line of last year's cases. Cases started earlier, peaked higher, and lasted a touch longer than we had seen in the past 5 years, and we had really seen in 20 years. Especially early in the season when we started our mosquito surveillance and when we saw those mosquito numbers and how many were infected with West Nile, we knew that it was going to be a difficult year. I also like to use this to throw into perspective with West Nile virus and, with a number of vector-borne diseases, we don't have a vaccine. We also don't have a treatment. So our best bet is preventing human cases. And to do that, it's a lot of surveillance work. So with the big picture of the point of surveillance is to prevent human cases of disease, why else do we do surveillance? What is important about doing surveillance? And I want you guys to chime in here. There are some hints on the screen that might nudge you. But what are some of the end goals of vector surveillance?
You might want to identify if a new species is moving into the area.
Bingo, exactly. Watching out for invasive species. The two vectors in the bottom right corner. Does anyone know what these two are?
That's the Lone Star tick.
Mm-hmm.
I think the other one might be Aedes aegypti.
Bingo, nailed it. Yes. So both the Lone Star tick and Aedes aegypti We typically do not see in Colorado. We don't consider those endemic. With the Lone Star tick, ticks are really good hitchhikers. So in our tick surveillance, we occasionally see those introduced to the state. Thankfully, we haven't found them locally yet. With Aedes aegypti is starting to creep through our region and approaching western Colorado. Southern Utah has had a challenge with that, and we will likely see it in western Colorado next. So we want to make sure we are doing surveillance so that we spot those invasive species before we have to deal with the diseases that they transmit so we can react accordingly. Great. Why else do we do surveillance?
I can answer. I think another reason for this is because we kind of want to be proactive and not reactive. We want to know like what's going to pop up and when it's going to pop up so that we can, instead of trying to, after people are already getting cases, it's kind of too late
Mhm.
in a way.
Absolutely. Yeah. As I mentioned, there is not a vaccine or treatment for West Nile. So we want to detect it in mosquitoes and try to stop it there before it gets into human cases. So a big part of that is helping inform our control efforts. When we start to see certain types of mosquitoes, especially when we see that they are infected with West Nile, we want to respond accordingly with going out and either controlling the adults or controlling those larval populations before they can even become adults to interrupt that process. Right. I can think of one or two other reasons why we conduct surveillance. Anybody else want to chime in?
I feel like it's important for the public to have awareness on what's going on in your area and then inform the public to change their own personal behavior to protect themselves.
I can't hear you very well, Tatum, but I think you were getting at public messaging. Yes. So absolutely. We not only want to get out there and control the mosquitoes, but we want to make sure the public is informed. A lot of mosquito prevention is up to the individual, to making sure they're careful when they're outside, they're wearing the proper gear, using their insect repellent. So we want to not only message that to them so they know what to do, but let them know when that is most important, when we are seeing those important species and when they are infected with West Nile. Great. So that is kind of all of the pieces that go into our application of vector surveillance. Really quickly on the left-hand side here, we've got two different types of mosquito traps. We've got the CDC light trap and a gravid trap. I just wanted to point out here, for mosquito surveillance, most of our traps follow a pretty simple premise. You need some kind of bait. You need a fan to make some airflow to pull the mosquitoes in, and you need a net. In the CDC light trap, our bait is primarily in that cooler, is dry ice. And as that falls and leaks out of the container, it distributes CO2. The mosquitoes sense that, think they've found a nice blood meal. They come in host seeking, and instead down here, they get pulled into that net. The light as part of the net also serves as some bait, though they're starting to see some evidence that mostly increases other insects getting into the trap. It seems to be more of an attractant for non-mosquitoes than actual mosquitoes. So some mosquito surveillance districts are starting to eliminate lights from their CO2 traps. The gravid trap is a little bit different. In this case, the bait is really stinky water. So we take water and add some hay or grass or other plants. Sometimes people use a little bit of like animal food, and you let that sit and ferment and get nasty. You add that to the bottom of the trap, and mosquitoes think that's a great place to lay their eggs. So a pregnant female mosquito, or gravid, which is the name of the trap, will come in, and instead of getting to land on the surface to lay her eggs, she gets pulled up into the trap. So both traps have a bait, a fan, and a net. However, our CO2 light trap, or CDC light trap, targets mosquitoes that are looking for a blood meal, and our gravid trap targets mosquitoes that are looking for a place to lay their eggs. So during surveillance, it's important to realize what mosquitoes you might be targeting, not only what species, but what life stage, what kind of behaviors you are attracting. Once we have those mosquitoes in a trap, we get to identify them. Some of you this summer will be involved in this process. I did a ton of this through my master's and PhD program. So I won't be going into full anatomy details today, but for what I will be talking about, let's look at first the head of the mosquito. Off the mosquito's head, we've got its proboscis. That's its long needle nose. These are its feeding mouth parts. Just outside of that, we have what are called the palps. And just outside of those, we have the antennae. The palps and the antennae are kind of sensory organs for the mosquitoes. They use that to not only find sources of blood meals, but also to find mosquito mates of the opposite sex when they need to. Now we're going to jump down to the abdomen of the mosquito, specifically the very hind quarters. So the abdomen has these different segments along the way. And one of the things we look at when we're identifying mosquitoes is these last few segments. Do they taper off sharply and come to kind of a point? Or do they end really abruptly and bluntly in kind of a rounded butt? I'll circle back to that with some more specifics in a minute. If you were taking a medical entomology course, or were doing broader entomology where you really had an unknown sample that you needed to ID, you'll be looking at so many parts of this mosquito. This might be the patterns on its thorax. This could be the relative lengths of these veins on its wings. You might be looking at clusters of hair on the sides of its head, or the coloration patterns of the different segments down its abdomen. So when you get full training in mosquito identification, you'll be looking at all of this in much closer detail. Has anyone here already started using a dichotomous key or have some experience in that? Okay, we've got a couple of people, great. So for those that haven't, a dichotomous key walks us through how we identify a mosquito. And what it does is we look at one more of that mosquito, and it gives us two choices. So part of a dichotomous key will look like this. Importantly, most dichotomous keys are for female adult mosquitoes. Only the females blood feed. So that is what we are most interested in for both pathogen testing, and that's where we want to focus for identification and control. And a lot of keys are also going to be regional. By using regional keys, we narrow down the possibilities, and that makes our key a little more focused and a little bit less massive and confusing. So this specific key is one I used in graduate school a lot. It is the female adult mosquitoes of Texas. And as I mentioned, throughout a key, as you're identifying a mosquito, you're going to be looking at all different parts of it. Different clusters and patterns of hair, the proboscis, the wings, all of the scale patterns, the abdomen. If we take a closer look at the top of this key, our first question is, how long are those palps relative to the proboscis? So we've got our proboscis mouth part right down the middle, the palps just outside of that. If those palps are almost as long as the proboscis, that tells me immediately this is an Anopheles mosquito. Since for West Nile virus, we're focusing on the Culex mosquitoes that transmit it. Once we see Anopheles mosquitoes, we would set those aside. We would not send those in for testing. If it's got really short little palps, that's a non-Anopheles mosquito. All right, next question. This one's kind of a neat one. The proboscis, is that straight, or does that sharply curve down like on the left? If it sharply curves, that's actually a Toxorhynchides mosquito. These are some of the very few mosquitoes that don't blood feed as adults. When Toxorhynchides are larvae, they eat other mosquito larvae, sometimes each other, and other insects that might be in the water. So as adults, they don't need that protein from blood meals. If we've got a straight proboscis, then we go down. The next question is about relative lengths of veins on the wings, and down we go following this key. I'm going to give you guys some quick shortcuts today to identify our species of public health importance, especially in Colorado. So as I mentioned, one of the first things we're going to look at is the palp. We'll use the palps and the antennae to both identify the male mosquitoes that we don't want to test and the Anopheles mosquitoes. Anopheles are relatively rare in Colorado, very important globally. So if you're doing surveillance here in Colorado, you might not see Anopheles this summer. You might see a few of them. It's important to be on the lookout anyway. So if they have very long palps and very bushy antennae is key, that's a male mosquito. If you were sorting through your pile of mosquitoes from a trap, first thing you want to do is just get rid of all of your males. Those antennae are very bushy, easy to spot. Long palps tells us that this is either a male, or on the right-hand side here, we see, again, an Anopheles mosquito. This one has short bristles on the antennae, so this would be female, but those long palps tell us that this is Anopheles. In the middle picture there, we have non-bushy antennae and short palps. That tells me that this is a female non-Anopheline mosquito. So once we have short palps, not bushy antennae, we would take those mosquitoes and continue through our identification process. Then we go from looking at the head, as I mentioned, to looking at that butt. On the left-hand side, we see an example of an Aedes mosquito. Specifically, look at those last few segments on the abdomen. They don't come to like a sharp point or a stinger, but they taper off more dramatically. And we think about that as a point. On the right-hand side, we have a Culex abdomen, and its last segment or two on the abdomen is going to end bluntly and rounded. This can be a little bit tricky at first, especially depending on the shape your specimens are in. If you are doing mosquito surveillance this summer, this will be one of the things that once you start practicing, you'll get better at IDing it. And it's sometimes not as obvious as the pictures, but thinking about a pointy A for Aedes and a rounded C for Culex helps me remember the difference between those two. Once we have our Culex female mosquitoes, we know that those are our potential vectors of West Nile. So as you're sorting those out, that is what would be sent into the state for West Nile virus testing. If we wanted to identify specifically our most important vector, that would be Culex tarsalis. So another important point with keys and working through the identification process is the order is there for a reason. You don't want to jump straight to the final identifying feature you're looking at without first sorting out the blunted abdomens to make sure you're looking at Culex. So if you know you're looking at a Culex, take a look at its proboscis. The tarsalis mosquitoes have a stripe of pale coloring about halfway down the proboscis. And this makes them unique among Colorado Culex. So then we know we're looking at tarsalis. We can also look at its legs. Sometimes mosquitoes, especially coming through a trap, right? They've been pulled through a fan, they've been trapped in a net overnight, they've been banging into each other and the net. They might be losing some legs, they might be losing some scales that we use to ID them. Sometimes it's important to have redundant features to identify a mosquito. In this case, we also look at the legs, especially those rear legs have this nice banding pattern where the joints of tarsalis have, again, that white or pale coloring down the leg. But if you skipped looking at the end of the abdomen first and jumped straight to looking at those stripe patterns on the proboscis and on the legs, you might get some Aedes that have a similar coloration pattern. So again, don't shortcut to the very end. You want to follow that step by step for a reason to make sure you're looking at the right mosquitoes. So once we have our pool of female Culex, or hopefully even female Culex tarsalis, those get added to a tube. When we do testing at the state and we send this out to health departments, that tube gets preloaded with our little BBs. They simply add mosquitoes to the tube, tell us where the trap was and what species they identified, and then send it back to our lab. Our lab adds the PCR reagents to that tube, shakes it up into a nice mosquito slurry, and then that slurry gets tested by PCR for West Nile. On the right here is a part of the mosquito surveillance that we did last summer. So remember all of those terrible human cases. We tested about 2,000 mosquitoes and just shy of 2,000 mosquito pools. And about 200 of those were positive. And 10% positivity might not sound like much, but when it comes to finding infected mosquitoes, that's pretty high. So we knew that this was a pretty bad year. And every county that we tested, Mesa's data is missing there, but every county that we tested detected at least some West Nile virus activity, which is unusual. Sometimes counties don't detect any West Nile virus activity in mosquitoes. And if you look at Boulder, for example, about a quarter of their mosquito pools were positive. Boulder had a particularly nasty year. Larimer on this chart stands out as doing way more mosquito surveillance than anybody else. A big part of that is because of their collaboration with Colorado State and the EBLE lab. Since the EBLE lab is able to do testing through their lab, they're able to do much more than other counties in the state. We are working on, through RaHP VEC and other funding mechanisms, looking at other ways to support health departments in other parts of Colorado so that they can have maybe not similar testing levels, that's really high, but at least to be able to do more than they're currently doing. And this is not only enabling them to do more mosquito surveillance and collection, but also, like a lot of things, unfortunately, this comes down to how much funding is available and how we can distribute that funding for West Nile tests. Okay, switching gears over to ticks. Again, not going to go into every detail of tick anatomy today, but I did want to show you on the dorsal view of the tick, the backside, this scutum here. A scutum like this that takes up about half of the tick's back tells us that this tick is a hard tick and it is female. On the male hard ticks, that scutum will take up the entirety of the back. And if that scutum is absent, we're looking at a soft tick. We very rarely see those in surveillance because they tend to bite and leave the host quickly. Hard ticks bite and stay attached for hours or days. So those are the ones that we usually find. When we flip that tick over, just like some of the first things that we look at in mosquitoes are whether it's male or female, and whether it's anopheles or non-anopheles. Both male and female ticks blood feed, so we don't care as much about sorting that out. But we do want to determine if that tick is Ixodes or non-Ixodes. Ixodes ticks transmit Lyme disease, which is the most common vector-borne disease in the US. Thank goodness we don't have Ixodes ticks in Colorado yet. We're on the lookout for them because, again, they're very good hitchhikers. They're expanding their geographic range. They will likely be here at some point in the future, and we want to catch them first before we see human cases of Lyme disease. So, unfortunately, to detect Ixodes versus non-Ixodes ticks, I'm going to draw your attention to the anus and the anal groove. When you've got the tick flipped over, you're going to look at that very back end of the tick. And on the left-hand side, if that anal groove is very evident and opens up towards the back of the tick, that is immediately an Ixodes tick. If that anal groove is behind the anus and opens up towards the front of the tick, that's a non-Ixodes tick. And that is one of the first things I look at when I am sorting ticks. So briefly, what does tick surveillance look like throughout Colorado? For mosquito surveillance, we set out a trap with bait and the mosquitoes come to us. That doesn't work as well with ticks. So normally we come to them or we find them already attached. So on the left-hand side there, we see a couple of people doing tick dragging, where you simply drag a light-colored cloth along a trail or along an area where you believe there are ticks. They like brush and long grass and shaded areas, relatively high humidity. They sense that movement and the CO2 and other signatures coming off of these guys, think they found a nice blood meal. They grab onto that white cloth, then they're easy to spot afterwards, pick off, and identify. This requires a little bit of training and a fair bit of time to go out and do this surveillance regularly. So a lot of states either supplement or instead of that, use passive tick surveillance, where people or veterinarians or doctors who find ticks can send them in for identification and possibly testing. That is what we do here in Colorado. So veterinarians or just members of the public that find a tick on themselves or a dog or just in the environment as they're out and about can scan this QR code, bag the tick, pop it in an envelope, and send it straight to my desk. And then I identify all of those. Right now, we don't have a mechanism for testing pathogens, but we're working on some collaborations to supplement our surveillance with pathogen testing, which would be great. Last year, I received almost 400 ticks from around the state, and the vast majority of those were either the Rocky Mountain wood tick or the American dog tick, both of which are dermacenter tick species. And this is what that looks like when it gets to my desk. I like calling it my fan mail to make me feel slightly better about coming into envelopes full of ticks. But those ticks get double bagged. Most of the time, they're not engorged. Sometimes they are engorged and they are very big. And I simply toss those under my microscope, identify them. I put that in our database and I report that back to the submitter so they have an idea of what they sent in. Okay, the last thing before we go to questions from you guys. If you've already seen this in a presentation of mine, no cheating. But in Jurassic Park, the movie starts with, they found a mosquito that had fed on a dinosaur. That was trapped in amber, perfectly preserved. They used the blood from that mosquito to clone all of the dinosaurs in the park. Terrible science. It's garbage. The mosquito is bad. What is wrong with this mosquito?
Is it because its proboscis points down, which means it's
There
the
you
only
go.
one that doesn't blood feed?
Bingo. This is a Toxorhynchides mosquito. They don't blood feed. There was no blood in this mosquito. Jurassic Park could have picked just about any other type of mosquito and it would have been viable, but they picked the one wrong one. They picked this mosquito because its nickname is the elephant mosquito. These things are huge, so it's much more evident on screen, but they maybe should have consulted an entomologist first. So with some entomology training, you too can ruin your friend's favorite movies. Now, if you guys have any questions either about my career path, what I do about vectors and vector-borne disease surveillance in Colorado, we've got about 15 minutes left, so throw them at me. Yeah, Caitlin.
I have a question just about ticks in general.
Mhm.
I put it in the chat as well, but are they mostly just generalists, or do some of them actually have like host specific, they're looking for certain species?
Very good question. So with all vectors, that's important to consider because that kind of also tells us what diseases they would be good at transmitting. Using West Nile as an example, West Nile starts in birds. All infections start with an infected bird. A mosquito feeds on that bird, becomes infected. And then if they then feed on, a human or a horse or some other mammal, they can transmit that virus. If a mosquito was just feeding on birds or just feeding on mammals, it would be a very bad West Nile vector. For ticks, the biggest thing for ticks is that changes throughout their life cycle. When they are larvae and nymph, they are tiny little things and they like to feed on much smaller animals. Often this is small rodents. That tends to be where they pick up things like Rocky Mountain spotted fever, Colorado tick virus, Lyme disease. They stay infected throughout their life cycle. And then when they're adults, they go looking for larger blood meals. This could be a human, this might be a deer. And then they transmit that to us. Within adult ticks, there's also some host preferences. So absolutely, all vectors have some kind of host preference, and that is very important in how they transmit diseases.
Okay, thanks. And then my
Mhm.
second question I have is, how did you decide you wanted to work for the Department of Health versus like the CDC or like a government versus private or state?
Yeah, good question. I I was looking at both. So I wasn't looking as much at private companies or mosquito abatement districts because that's more of the actual surveillance work. And again, through my education, found that my brain's happier on that bigger picture. So I wanted something that was public health focused, that broad scale, and something where I would get to engage with the various partners. I love that I get to work with the mosquito abatement districts and the academic centers and have some work with the CDC. Honestly, if I had found a CDC fellowship that worked out, I very likely would have ended up there. I found I really enjoy being at the state level because I get the big picture, but I also get to work with those that are focusing on the details. And at the CDC, they are one step further big picture. And so it's a much larger scope. Yeah, we got another hand raised.
Yeah, I had a question. If we have a decent amount of Culex tarsalis and non-tarsalis, would you want us to limit the sample we send in to only tarsalis?
Depending on how many you have, yes. So let's say you've got enough to send in like a full tube of tarsalis. Focus on those. Those are our most important vector in Colorado. If, especially early in the season, there tends to be more pipiens, and you have enough to send in a sample of both, and you have the capacity to do that, then send them both in.
Okay, thank you.
Yeah, Maddie.
Yes, hello. Question for you. You mentioned you worked with the different counties across Colorado to sort of collaborate on mosquito and tick related issues. How do you navigate differences or even challenges amongst different types of work done amongst the counties? Or do you run into difficulties?
Yeah, so that's a really good question, and it's absolutely true. There are big differences. It's a matter of staying flexible and finding approaches that work well for a county's focus and capacity. A really good example is some of the counties along the Front Range versus the smaller counties in our Western Slope and Southern Colorado. So along the Front Range, there are a few counties that kind of contract out their surveillance efforts to a company called Vector Disease Control International, VDCI. So they pay VDCI, sets out their traps, collects their mosquitoes, sends them to the lab for testing. Out in Delta County, I just worked for a couple of days with their health department. They don't have that kind of funding. And they are actually pushing to expand for more vector surveillance and control efforts in their county. And the way they're doing that is through a lot of public engagement. So in their site, they actually have a few of the townships participate in their surveillance, and local community leaders can find either themselves volunteer or find volunteers to set out the traps in the different parts of the county, collect those, and either set them out for a pickup by the health department or take them to the health department itself. So absolutely, we do have to, stay flexible, find an approach that works for each county. And that's been a learning process in this role for sure. In Southern Colorado, a few counties in the San Luis Valley area are starting to do mosquito surveillance for the first time this year. So last month I went down and did some training with them, and they're starting out really small. They aren't even sure in which portions of the county they have Culex or Culex tarsalis. So that's where they're starting. And they're also very focused on public engagement, using volunteers to set out and collect their traps for them.
That makes sense. Okay. I also have one more question for you, a little bit off of what you answered previously, about the specific timeframes for collecting certain mosquito species. So you said that there are certain times we might see more tarsalis than pippins. Do you know what that range of months or weeks is?
Yeah, so with the caveat that there's a little bit of variation, it depends on the weather each year. Normally, the first thing we see once it starts getting warm enough, which is sometime between March, April, May, the first thing we'll see is Aedes mosquitoes. And that is because they lay their eggs in the fall and then they overwinter as those eggs. So as soon as there is enough water and warm enough weather, they hatch out in mass. So if you were getting eaten alive by mosquitoes about a month ago, those were very likely Aedes vexans that don't transmit human disease, but they earned that species name for a reason. They are very annoying. So they hatch out first because they're hanging out in the winter as eggs. Culex mosquitoes overwinter as adults. They lower their metabolic rate and find a relatively stable environment that might be like underground, in a wood pile, in a tree, depending on the species and their environment. So when it warms up, they have to then lay their eggs, and their population then builds up over the course of weeks and months until they're actually a problem. Because of differences in their behavior, we typically see pipiens first. So over the last month or so, as we've started seeing Culex, some regions see pipiens first. And then sometime as we head into June, those populations shift into seeing more tarsalis. That also varies across the state, depending on what area you're in, but that's the general trend we see. Aedes mosquitoes, pipiens, and then tarsalis.
Okay, awesome. Thank you so much.
Of course.
I have a question, I guess, Chris.
Mhm.
With the, I guess, mainly, I guess, just most generally, like, does identification like influence control decisions, and maybe in what way would, like, save for like the difference between, I guess, the top level, like finding Aedes versus finding Culex, or finding tarsalis versus pipiens, like how would that influence maybe different choices that would be made by the district?
Yeah, great question. So part of that is whether or not we are concerned about diseases. Obviously, we're finding a lot of Aedes. We aren't worried about West Nile transmission until those populations shift into much more Culex. We would also think about when and where they are biting, and where they are breeding. So Aedes, for example, tend to bite during the daytime. So if you are targeting those adults with some kind of intervention, you would have to be doing that during the day. At nighttime, they're much less active, and you're not going to be able to hit them with adulticide sprays. Culex tend to bite dawn and dusk. So if you are spraying adulticide to hit them, you want to be doing it like just before sunrise or just after sunset. They also have slightly different breeding behaviors. Aedes are floodwater mosquitoes. So that sounds like they're going to be in big plains that flood. You can find them in grassy fields that flood and recede, but they're also going to be in a lot of containers, plant pots, bird baths, things like that. So some of those are hard to target with large scale vector control, and you would want to make sure you are communicating to the public to make sure they are emptying and checking those containers in and around their home. So absolutely, the species that we find do influence our control efforts.
I got a question.
Mhm.
Is there concern at the moment for like introduction of the encephalitic viruses into Colorado, like St. Louis, and then the equine encephalitic viruses?
Great question. That's something that we keep an eye on. But most years we do not see them. And rather than an kind of an emergence and expansion of the geographic range, those tend to happen in kind of epidemic years. So they might show up and wreak havoc for a year, and then we don't see them again for several years. So we do keep an eye on that. There are some of the labs that do testing at the state level. We're focused on West Nile. Some of these smaller labs like mosquito abatement districts or academic centers that do testing test on a panel. So for example, if they got a hit that was positive for Western equine, then we would respond accordingly and change our testing approach at the state level. Another trigger for that might be human cases. So if a human comes in, a lot of those viruses present very similarly. So we'd likely test for West Nile. If West Nile was negative, you would expand that differential to include some of those other viruses. And if we get a positive human case for those, we would also certainly respond accordingly. All right. Well, if there's no other questions, I'll go ahead, Tim.
Question for you, Chris. Kind of on the different vein, I guess. We did a thing at CSU near the end of the semester where all the PIs went up and talked about their failures. Curious, like the way that I think it's often presented your career path, you know, like you went bounce from here to here to here. I wonder if you could just touch on like, were there any moments that were maybe discouraging, or did you like deal with rejections? I know you said you were applying at CDC and stuff. So I think from the finished product side, it's like, oh wow, you were just finding, you know, everything worked out. But I imagine that there were some speed bumps in there. If you just want to give.
Yeah, absolutely. Two that I kind of touched on already was my my dissertation project. The goal was to focus more on virology and the human side of things. But then Zika showed up and forced me to change my plans and further specialize in mosquitoes. That might less be a failure, but an unexpected turn. My postdoc as well, supposed to be doing a lot of field work and doing different intervention strategies. And unfortunately got stuck in the lab. And again, maybe not a failure, but an experience that I learned from that was very unexpected. During my postdoc, I applied to the CDC Epidemic Intelligence Service, the EIS fellowship program, and made it to the interview stage, and then was not offered a spot. So that was a little bit discouraging. A lot of people that I talked to along the way said they don't take a lot of people with an entomology focus. And that might vary year to year depending on what they are focusing on. But that was a little bit frustrating. Going even further back from that, during my undergrad, I did a summer internship with the state health department. Rather than mosquitoes, I was doing HIV and other STDs, doing some surveillance work and some outreach. And I really did not enjoy that. I really liked the outreach part of it. But the number crunching and the people that I worked with and that environment was not for me. And I came out of that thinking like, all right, well, lesson learned. I don't want to work at a state health department. So it's important that in, you know, academic and public health settings, so much depends on your role, the project that you're working on, especially the team that you're working with. I'm really happy at the state level here in Colorado. But the experience I had at that time was, again, not for me. Caitlin, you have another question.
Yeah, my question was, I don't know how much experience you have with this, but just trying to complete a project, how do you go about trying to be clear with other people that you want to include in your group? I've come across people that are like really protective of their data or very protective of what they, like if it's not published yet. How do you go about being like, I don't want to take your data, I'm just trying to like add to it or create a project out of it?
Yeah. Honestly, be direct, set expectations as well as how that data is or is not going to be used. And if they are, you know, hesitant to share data for something you think is important, try to outline why you think it's so important. Good example I'm dealing with right now, actually. We are working on getting our data put through a different pipeline called VectorServe. This will do some analysis for us. It'll, you know, be great for mapping data throughout the year. But that requires collaborating between, you know, the data that I have access to, what the counties are contributing, the mosquito abatement districts that have a separate set of data, the private corporations that get contracted by towns or neighborhoods to do surveillance, and academic centers. And mostly everyone's on board. The challenge is that private sector. They tend to be more protective of their data. So it's been finding a middle ground with them, the data that they are willing to share. But they wanted to know specifically who is going to have access to that data, how it is going to be communicated to the public, how their name will or won't be attached. And it's taken multiple conversations to get to where I think they are on board. Yeah, so be direct, set expectations, address concerns. With conversations like that, I think it's best not to beat around the bush. Does that
Okay.
answer your question?
Yes.
I think that puts us just about at time. So thanks, Chris, for the presentation. Thanks everybody for coming.